Welcome to a LabRoots webinar hosted by QIAGEN and sponsored by QIAGEN. Today we have an expert panel with a really interesting discussion, powering the precision in precision medicine, the role of curated data, databases, and knowledge. My name is Dominic John. I lead up the marketing and the clinical product management team at QIAGEN Digital Insights. I'll be your MC today. If we look at the topic of today, I want to really question, how do we enable precision in precision medicine? It was interesting, in November, I got to see a report from a survey sponsored by Genomic. The interesting sort of factor there was we're asking molecular pathologists who were doing next generation sequencing, what was their confidence in interpreting their patient results here, looking at those variants from their NGS tests. And what really sort of shocked me and surprised me was the fact that over 45% of those who asked said they rated their confidence in interpretation less than 7 out of 10. To me, that means we've got a long way to go really to deliver on that precision medicine. And one of the big questions is, where is that confidence? And we've seen some interesting results as to what was driving that confidence. And I think a large part of that is access to high quality knowledge. And what we want to do today in this webinar here is use our expert panel to help the audience here understand some of the emerging challenges in genomic testing for somatic and hereditary diseases. And then, very importantly, let's look at the role of curated knowledge bases to support that and improve that confidence and deliver that precision. But recognizing at the same time, there's lots of different knowledge sources out there, different databases. They're curated in different ways, and you have a lot of choices. So this webinar will help you, and the panel will help you work out what are the right sources of information for you. So let's introduce our panelists here. We've got 3 great panelists. They all come with long experience in the industry here, building databases, presenting databases that support our next generation clinical interpretation. So maybe what we'll do here is just have each of the panelists introduce themselves, give a little bit of background, and then what I'd like to do is each of them talk about their products and their subject matter expertise here. So Lisa, why don't you go first? Why don't you give us a little bit of background about yourself?
Sure, thanks, Dominic. So I'm Lisa Barrow-Laing, and I am a senior application scientist here at QIAGEN. I have more than 15 years of experience working in the field of oncology and rare diseases. And I spent over 5 years as a variant scientist generating clinical reports. So currently at QIAGEN, I do serve as a scientific liaison, frequently delivering workshops, trainings, and demonstrations on our informatics solutions to directors and genetic counselors. And I also can assist with serving as a consultant, really, to help our customers understand what is the best secondary and tertiary analysis solutions to support their various tests, including hereditary cancer panels and clinical exomes.
Fantastic. Thanks, Lisa. Simon, why don't you give us a bit of background on yourself?
I'm going to sound fairly similar. I'm Simon Forbes. I'm at the Sanger Institute with the delightful title of Head of Cosmic, which means I run the Cosmic product and project at the Sanger Institute. I've been in cancer 5 years or more, both at Cambridge University and the Sanger Institute. And we're really looking to make sure Cosmic doesn't just detail all of the mutations in human cancer, but really makes that data accessible and easy to use and really very valuable in the pursuit of precision oncology.
Fantastic. Thanks, Simon. And then Shawn.
Hi. Hi, I'm Shawn Bauer. I'm a Director of Content Engineering at QIAGEN. I'm responsible for two main teams, the team of scientists who work on curation and ensuring that the content that we curate gets goes into our applications and is used appropriately, and then a group of knowledge engineers and bioinformaticists who work on the knowledge modeling and ensuring that process flows smoothly. Prior to that, I worked in drug discovery in a variety of areas. So it's an area I've been familiar with for quite a while. on different, two different sides, one on the drug discovery side, and now the use of the drugs in clinical applications.
Fantastic. So, from the audience perspective, we've got three, great panelists here with deep experience in curating, building knowledge bases and providing knowledge bases across both the somatic and hereditary domain, both in, I should say, research and the clinical setting here. So to sort of move the conversation forward here, I'd like each of those panelists to introduce their products and their line of expertise. And then after that, we'll go through some questions here around some of the trends they're seeing, some of the challenges that people are addressing, and then how does the content and what to look for in the content here. So maybe Lisa, why don't you sort of lead off here with your background in the products that you represent?
Sure, absolutely. So today I would like to just share a bit about Human Gene Mutation Database, or HGMD Professional. We have, been selling this product for an extended period, but there's a lot that I would like to share with you about HGMD. So essentially, if you're doing any type of hereditary interpretation, whether for clinical cases or in the research arena, you're probably familiar with HGMD. Because it really is considered to be the best tool for answering questions around, what is the impact of this mutation in terms of causing hereditary diseases. HGMD is curated explicitly from peer-reviewed published journals. So we do have a team of scientists that are manually curating the information from the literature, and it's truly an expert curated database. It only contains germline mutations, so you would not see any somatic mutations there. And I think while it is extensively used, what is really it is renowned for is that it is a gold standard. And that's because of the quality of the content and the rigor of the QA that has gone into this work to ensure that it is indeed accurate and consistent. So I used HGMD extensively. I was a variant scientist for about 5 years. And in the clinical lab, it was certainly the go-to for us to detect alterations and help to understand the causal variants in rare disorders. And what we're hearing our customers say is that they're saving 50% of time when they use HGMD compared to methods that they were using prior to adopting HGMD in their laboratory. So in terms of the curation source, we are partnering with the Institute of Medical Genetics at Cardiff University. They have established a database over 25 years ago. So again, just having that extensive experience in doing germline curation. So in terms of the publication rate, as scientists, we're very aware that, every day there are, hundreds of publications that are released. So being a variant scientist, it's very hard to figure out what is the pertinent publication for any genes or variants. So HGMD is able to save you time because it has this comprehensive database whereby you can just go to HGMD, readily search for a gene or for a variant or a phenotype, and be able to take advantage of the more than 127,000 articles that have been manually curated in HGMD. Additionally, our genetic experts or scientists are highly trained. Many of them have been with the company for the university for more than 5 years on average. So they're really very familiar with getting to the key findings from the publication and ensuring that it is accurately curated. Additionally, we are doing quarterly releases of HGMD. And what that does for you is that it really ensures that you have up-to-date content. So as of now, if you can see this graph here, you can see that, over the last decade or so, there's a steep incline here in terms of the amount of disease-causing mutations that have been cataloged in HGMD, with now more than 300,000 mutations being available. So we wanted to discuss a bit about the uses of HGMD. So we did do a recent survey. And we found that, if you were to use Google Scholar, there are more than 17,000 published studies that have referenced HGMD because they've pulled key information and identified causal variants from using HGMD. When we asked our clients what are the key uses and use cases, they shared that HDMD is very relevant in terms of being able to help them to search for mutations, to search for genes and diseases, but also being able to enable them to do advanced searches across different mutation types or characteristics. And then because we do have availability for the download option, many of our customers are actually downloading the data from HDMD and incorporating it into their own analysis pipeline. Additionally, they've found that with HDMD, they can readily design different panels of interest, and they're able to as well prioritize mutations. So it is really a robust database that can be used in many different ways to really help you to get to an answer or to, again, build out those internal pipelines. So I just mentioned the ability to download. We do have that available. I just want to differentiate HDMD because as an application scientist, I often will hear from some customers, isn't HDMD free? So we do offer HDMD public, which is free to academic users. And so again, having been in grad school, you might have heard of HDMD, even used that publicly available site. But the issue there is that that content is 3 years away from where we are with the current standard. So in order to get the most up-to-date content, you would have to subscribe to HDMD Professional. And there you can go to the web application and conduct those searches and queries, again, on the gene level, the alteration level, or diseases. But you also have the ability to be able to download with another subscription the content from HDMD Professional. So this may be a little bit small, this table, but it's really just going over several of the different types of searches that are available for you. Essentially, the key point here is that for public, you have a limitation in terms of the amount of search and queries that you can perform as compared to HDMD Professional, where you can see you do have a full functionality across the board to search across various different attributes, whether you're interested in searching on variant classes or various ontologies or understanding genome sequence information. That's all available to you in HDMD Professional. So with that, I'll pause there so we can discuss some of our other databases.
Fantastic. So great background there, Lisa. Let's move into Simon to give us a bit of background on the somatic side. So Simon, if you want to just lead away.
So I'm going to present COSMIC. COSMIC, in a similar way to HDMD, is a genotype-phenotype relationship database across the somatic mutations in cancer. So we're focused on human cancer, and we're focused on the 90% of mutations that drive cancer by acquisition across a lifetime's exposure to a variety of carcinogenic effects. And the idea in COSMIC is similar to HDMD. We will capture information from a wide variety of sources, primarily the literature, but also the expanding range of international genome consortia producing huge genome-wide content across cancer. We'll standardize that in a variety of forms, integrate it and annotate it in standard ways, and kind of make that available in a wide variety of different forms for integration or analysis in a number of different ways. The data in COSMIC is curated primarily by hand using two related pathways. On the left-hand side, large-scale genomic data usually comes along with a human genome coordinate and can be annotated for genic consequence using a database like Ensembl. And the hundreds of thousands or sometimes tens of millions of mutations coming out of cancer genomes get annotated in that fashion. But the majority of very cancer gene-specific information going into COSMIC is hand-curated. We've got a team of curators who are experts in their field with previous scientific expertise, and they look at the literature for the genes that we know drive cancer, and they can tease out all of the detailed bits of information also, I froze for a second there, they're teasing out not just the mutations that are driving disease, but also the disease descriptions themselves at high resolution. And the idea is to not just be very specific about the mutation, but also the disease. And that's where you'll get the most significant genotype-phenotype relationships, which mutations are driving which disease. The broad genomic data from the big wide studies is merged with the real deep insights we get from these gene-by-genes assessments, and they form the basis of the COSMIC database. And the idea into COSMIC, you can immediately discover which mutations are significant in which disease. And I'm just comparing two visions of the KIT oncogene here. And on the left, you can see a fairly rare blood cancer, mast cell neoplasm, is clearly driven by a single nucleotide variant, resulting in a D816V change in the phosphotransferase domain. And on the right-hand side, there's a much broader range of mutations driving a gastrointestinal tumor. There's a little bit of D816V, but there's a much broader range of substitutions and also a much higher number of deletions. And we start looking at this scale of database in this resolution. You can really see which mutations are driving which disease, but also how they change across genes and across diseases. And of course, it's this information that's essential to which targets are appropriate in pharmaceutical design, which ones are useful and how do you characterize them, which biomarkers are important. Not just how do you annotate diagnostic reports, but how do you explore them from a VUS perspective? Which ones can you give some information if you don't quite know what it's trying to tell you? And looking at COSMIC and leading on from COSMIC, we released last year the cancer mutation census. This is a tier on top of COSMIC that details a wide variety of population and biological and other observation properties across each mutation in COSMIC. And this gives you a good way to navigate every single mutation in COSMIC from a variety of perspectives to not just identify which mutations we know matter in disease, but which ones have potentially oncogenic properties that allow you to navigate their uncertain status. If it's a VUS, a variant of unknown significance, how do you interpret it? And the cancer mutation census gives you lots of information to support that interpretation. And our latest release a few weeks ago included a new component of COSMIC on mutation actionability. And this is not just all the observations of mutations in cancer, not just the properties that allow you to evaluate which mutations are important in cancer, but this is a very strong list of which mutations are actionable, which mutations can you treat with which drug in which disease, and with what survivability metrics. This is curated primarily from the literature using follow-up studies from clinical trials and a variety of other literature exploring the consequence of treating various cancers with mutations with a variety of drugs. And this gives you really good ways of navigating how to apply precision oncology in a clinical audience. Thank you.
All right. Fantastic, Simon. So we've got Lisa representing the hereditary side with HDMD, and then COSMIC on the oncology. Let's hear from Shawn, who's actually got probably a bit of both, but let's hear.
Yeah, so let me jump in here. I'm going to talk a little more about the focus of this will be the QIAGEN knowledge base, which is our repository of information, which then goes out to power our content-based applications such as IPA, QCI, QIAGEN Clinical Insight, Interpret. And that would be both the hereditary and somatic workflows. So our curation process is this is similar in some ways to what Simon described. So at a high level, we start with unstructured data and then also structured data. For unstructured, that would be we'll need to identify articles that would be expected to have content relevant to our applications. These are primarily articles from the primary literature. can also be clinical trials, drug labels, clinical practice guidelines, and things like that. These are then manually curated using our trained scientists and our curation tools, and then imported so that we've then transformed that unstructured information from those documents and other sources into information that's structured and thus computationally accessible. For structured imports, that's a little bit easier in some ways. We take structured information such as tables or XML, things of that sort from various sources, map them to the concepts that represent the same thing in our knowledge base, and then import those. Both of these are done as imported and updated as to keep the content current. As each subsequent release of a database that we've licensed gets updated, we then import that content into our knowledge base, and in many cases put it out as part of our weekly content updates. So the kind of the fundamental point behind that is to take unstructured and structured data and integrate it all within one common framework so that it can all be used essentially interchangeably. And that requires, the translation of the information in, transformation into our framework, of course, testing, looking for consistencies, inconsistencies, and of course, then export into our applications. And then once that goes into our applications, like Ingenuity Pathway Analysis or QIAGEN Clinical Insight, that's when it leads to, it then becomes more useful, I guess, for the customers. So one of the fundamental points of the information that we capture is its highly detailed nature and its how highly accessible those details are within our findings. So when our scientists curate information from these sources, such as here's an example of a drug approval from some time ago, where each of the elements of this finding are essentially mapped to concepts within our knowledge base, and then thus become accessible. So, for example, the drug has been approved to prevent skeletal-related events associated with bone metastases in a human with a solid tumor. Each of those elements then can be accessed and used in our applications, which has proven really useful in the space of QCI, especially around QCI somatic, as drug approvals have evolved and the uses of them have become more refined. There's more and more detail that we've been capturing along the way, which has proven useful for creating clarity for the various customers. So I think that's something that's going to continue to be a challenge as we move forward, as the biomarkers become more complicated, or the combination of biomarkers for a given indication, for example, as that expands and the various use cases expand, that richness serves us well. The scale of that content is quite large. We've been curating information into our knowledge base for over 20 years. That's resulted in well over 20 million findings. That includes manually curated information as well as the database imports. Just as a slice of that, we have over 33,000 various disease classes and subclasses covered. That results in over 1 billion edges within our knowledge base. This is comprised of over 850,000 references and greater than 35 integrated scientific and clinical databases. Our knowledge base contains over 3 million biological concepts, and as I said, we're doing these updates essentially daily, so we have greater than 5,000 new data elements and findings are imported per day. That content then goes out to our various applications along standard updates, and then is used by our users. That's it.
All right, fantastic. Thank you, Shawn. Just to sort of put some perspective there, Shawn, correct me if I'm wrong. I think the knowledge base here has been used to process over 2 million patient cases, genomic profiling.
Oh, sorry, that audio broke up just a bit there. Could you repeat?
Yeah, so I believe the knowledge base has been used to process over 2 million patient cases for genomic profiling to date. Is that correct?
Yes, I believe that's true. Yes.
Yeah. All right. Yeah. No. So just summary here. Sort of 3 experts here, 3 different sort of products that are delivering knowledge here. what I'd like to do now is really get into a Q&A session and start drilling down and extracting some expertise and some guidance from the panelists here. So really, I think, step back a little bit and, let's look at the trends before we talk about, the what and the how. But, let's step back and look at the trends that are impacting genomic profiling, clinical testing here. So to the panelists here, what do you see as the major trends in next generation sequencing testing? And then, how is that sort of affecting interpretation? So anyone feel free to answer.
Sure. So I would say, having worked with several hereditary testing labs over the last few years, what I'm seeing happening more over the last year is that more customers are interested in looking at copy number changes, so CNVs. That is something that I think NGS before, there were some limitations there, but more and more labs are, venturing into doing copy number analysis. And of course, once you have those detections, you need to be able to understand what it actually means to that specific hereditary cancer or specific disorder. So it's important more and more to also capture the structural variants and look at the impact of that from the literature. We are also seeing, I believe, more tumor normal testing as a norm in our clients doing those types of applications, as well as looking very closely at panels for cardiomyopathy and Alzheimer's as well. So that's something that I think is much more adopted recently, and we have a lot of curation for that in Hdmd as well.
All right, Simon or Shawn, what trends do you see?
Following on from Lisa, I agree that there's more to it than the DNA-seq. DNA-seq used to be the major way that it interpreted this information, but there are many more mutation mechanisms, especially in cancer, just looking at it from a cancer perspective, that are driving disease than anything a simple DNA-seq test can reveal. So while we've got in COSMIC, I think it's just over 30 million mutations, we've actually got 70-odd million somatic variants encompassing a wide variety of different mutation mechanisms. And just looking through the kind of thing that you see people navigating precision oncology by, if you have a lung cancer with an EGFR mutation, standard procedure would be to give a drug called gefitinib. But as the cancer evolves, quite often a T790M mutation results in resistance to that drug. Therefore, you need a new drug, something like osimertinib, and there's this kind of constant evolution that you can assess by DNA-seq, but that evolution needs to be watched. It's not a single one process anymore. And then beyond that, the mutation status of, or the inactivation status of genes is very important to know. If you have an ATM gene inactivation, you get a good response to radiotherapy. If you don't have that inactivation, then radiotherapy might not be the way to go with the patient. Similarly, if you inactivate BRCA, you get a good response to drugs that inhibit DNA repair. And there's a great way of putting together a number of different molecular mechanisms and mutations, all of which can appear independently, to begin to frame a really accurate picture of how to treat any individual patient.
All right, thanks, Simon. And Shawn?
Yeah, I absolutely, in the somatic space, Simon summed it up very nicely. In the time that I've been working on this, when we first started QCI, even, obviously before it was released, when it was in wireframes, there were only a handful of drugs that on the label had a biomarker as a prerequisite for using the drug, and gefitinib would be one of the very early ones. And then as time has evolved, there's multiple EGFR inhibitors, and then the way those get targeted and used, and then especially around patients that have undergone multiple lines of therapy. So they may start with gefitinib, then they go on to osimertinib, and then there's another change and another change, and tracking those things, and then and making, and then as people continue to do clinical trials and various other studies that get reported, understanding what is the, so in taking that cancer sample, that NGS sample, and also it's being tested in non-NGS ways, and taking all of that input from that describes the tumor and synthesizing it into one system where you can generate a report from that, and then taking the complexity of all that's changing within the literature and trying to come up with the right therapy or the right possible therapeutic recommendations for them. It's becoming increasingly complicated. So, anything that we can do to move that forward and make use of the information from the literature and present it to the clinicians, quickly and in a way that's easily understandable is going to be really valuable. And it's interesting. I mean, when we first started, the notion of biomarkers and combination biomarkers. So, the patient that has T790M plus L838, for example, knowing that that's they're sensitized to gefetinib, but it's also resistant. That's a very simple case, and I mean, that one's been around for a while, but that exists for many different drugs now, and it makes it very challenging. So
essentially, you guys all jumped on sort of the complexity of the cases. We're looking at precision medicine, and now there's all these sort of external pieces of information that help you with that new sort of decision, which goes back to that original sort of point of confidence. It's getting more and more challenging from that perspective. But you didn't comment on, panels and whole genome, whole exome. I mean, what sort of trends are you seeing there in terms of uptake of panel size and whole genome, whole exome? Do you want to comment?
Yeah, so I'm certainly seeing whole exome and whole genome sequencing being done more often. I think there are still, for some of our clients, issues around reimbursement. And so, it might be the case that virtual panels might be an approach that is used. Also, some of the panels that are available, when you start to look at the smaller panels for Alzheimer's or Parkinson's, it's not clear just from the vendors why those genes are on the panel. So back to the confidence of that, being able to use a database like Hdmd, where you can really query across the phenotype or disease and see all of the evidence just at your fingertips. That has been really helpful towards allowing our customers to have more confidence in the panel testing that they're doing. But clinical exome is something that is being onboarded more frequently, just again, with some of the complexity of, okay, how do I get this reimbursed? And then what does all the data mean? And that's why it's just so important to be able to leverage the content in the databases that we're discussing.
Yeah, Shawn, Simon?
Yeah, I agree. It's still fairly recent that we're having conversations with people on which 17 genes we should be testing across hematological cancers. 17 genes feels like a tiny number of genes to be testing these days, but of course, it's possible to recommend, for reasons of scale, you want to assay all 700 genes that may be involved in cancer. But then you're getting towards very large panels and exomes. And we do talk to people who are doing exomes and genomes, and there's a lot of discovery behind that. But of course, there's a lot of complexity in its interpretation. When you do a genome analysis of a tumor versus a normal, you'll find tens of thousands, hundreds of thousands, sometimes millions of mutations. Somatic mutations, most of which are a consequence of disease rather than driving the disease, and most of which aren't treatable in any sense whatsoever. And I think there's a slight fear in exomes and genomes that you're also going to find incidental findings. Is there something in there that might misdirect treatment? So having accurate information on what is significant in test design, what should I be looking for and what shouldn't I be looking for, is essential in navigating this really complex process.
Yeah, and Simon, I think you're sort of, just to reflect on the conversation here about the trends, it's really two things. We're seeing the diversity and the complexity of the decisions that have to be made. And the second one is, there's progression from these smaller targeted panels to larger targeted to whole exome. And obviously that creates a whole set of new challenges, especially when it comes to interpretation, precision medicine, and confidence. So maybe let's move to the second one. So from an interpretation, a confidence, precision medicine perspective, what do you see are the challenges that are now being faced by these labs or research organizations adopting NGS in the clinical world? Lisa? Or?
Yeah, definitely. I was pausing, but yeah, no, certainly, the clinical guidelines, right? So there are, ever evolving clinical guidelines that are coming out. There are some that are more standard, like the ACMG guidelines. But even recently, there was an updated paper on how to, for instance, classify those CNVs, right? And being able to not just read, understand, digest that information, but then know how to incorporate that in terms of, the identification of pathogenicity of a particular variant or whether it would cause disease, that can also be very, very challenging. So, it's really great that not only in HGMD, for instance, you have the ability to understand what that underlying evidence is and all of the references that support the classification of the variant, but, they do have very well structured ways of categorizing all of the variants so that you can get a sense for, what is actually causing a disease versus what is perhaps just a variant of unknown significance or uncertain significance. And additionally, in HGMD, it is really just germline mutations. So that's very helpful for hereditary labs that aren't distracted with other types of alterations that are not important for hereditary diseases. And beyond that, you don't have any polymorphisms. So I think one of the challenges that labs are facing is, how to really incorporate the guidelines so that they're sure they're accurately classifying the variants and then stay on top of all of the new changes that are coming out to the various guidelines as well.
Yeah, Shawn?
Oh, I think Simon was.
Okay.
You've been pointed out. I'll let you go first.
Oh, I would just, kind of following up on that. I think that especially as we move on to larger and larger panels, well, actually, when I see the larger and larger panels, I typically think of the content acquisition side, the challenges of doing that, making sure we have the appropriate content, and then, of course, providing it to the users in a way where it'll be, clear and understandable, and especially filtering through the variants that are going to have an effect and drive the disease versus ones that are passengers or less relevant. And so it's one thing to run a bigger panel and get more information, and that's obviously going to be very, very helpful, especially in areas where you are looking for maybe a rare variant which is leading to the disease, but then understanding how to take that variant amongst all of the others and understand that that's the variant that's the driver and that is relevant, making use of the information from the literature and any kind of prediction algorithms we can use. It seems to be, when I look at these, those are the challenges that I don't know, kind of make me a little nervous, let's say, because we'll have to, expand our content acquisition, find the variants in literature, but also integrate them in a way that makes them actionable, because it's, having more data is a good thing, but you have to, have an insight from that data and be able to understand what it actually means and what the next step would be as far as, predicting the risk of disease in the hereditary workflow, or, what the next therapeutic option is, or what the estimated, what effect it would have on the patient's prognosis. So. I don't know, but I'm a little less on the customer-facing and app side, a little more on the content and the engineering side. So my perspective, I think, might be a little different that way.
Simon?
Mm-hmm.
Yeah, definitely agree. I think it wasn't that long ago when people had a look of panic in their faces when you were having a conversation with them around navigating hundreds of thousands of mutations per genome. And the expectation was we'd get to large-scale NGS, at least exomes, to really understand what the genomics was behind any individual patient's cancer. But which mutations actually matter, rather than which mutations are there, is a real challenge. And as everyone's saying, there are various levels of uncertainty around much variant interpretation, and how much information can you bring to support that evaluation is so important now, because we can rattle off a number of mutations that we know drive cancer, and you can say KIT, you can say EGFR, you can say KRAS, but underneath that, if you're looking at between 5 and 10 drivers per cancer, with a continually evolving resistance mechanism to any individual therapeutic intervention, then there is a continual almost requirement on mutation interpretation. the more information you can throw at that, the better. And then, of course, trying to keep up to date with which drugs do useful things to particular patients with particular diseases, with particular mutations, is a big, complex challenge to keep up to date with. And I think beyond or within the three of our resources, hopefully we're really looking to provide solutions across the majority of those questions.
Mm-hmm. Yeah. So if you point to a really, point instead of, isn't just the volume of data, but also the timeliness of the data as well is key. It's interesting, personal experience here. I was talking to a clinical lab, and, they were saying, they've just moved up to a 500 gene panel for cancer, and they were taking, on average 6 hours a case to interpret. And, they simply said we can't scale. It was just the sheer volume of content and information that they had to go research before they felt confident in delivering that report. So really moving this on, maybe flip around. I've heard various pieces of this. Do you want to just summarize? Well, how do our knowledge bases? How do these knowledge bases help resolve that? I think there's some fairly short answers to this. Maybe Simon, do you want to
go? So people have been using Cosmic for years to at least annotate patient reports, and you can begin to say this mutation has been seen frequently, therefore it's likely to have some impact. But these sort of evaluations are more complex than that. So that's why we've been putting together more targeted bits of Cosmic, if you like, the cancer mutation census, really to add detail. Not just has this mutation been seen frequently, but has it not been seen in non-cancer populations? Has it been seen in disease-specific populations? What are its biological characteristics? Those sorts of things that gives you an idea that it's significant. And then, of course, beyond that, from a very clinical perspective, not just which mutation appears to matter, but which mutation can I actually do something about? I think that is getting to the crux of the problem, is which mutation can I do anything about? Because while you might have tens of thousands coming out of an exome, if you can't do anything about anything that looks significant, it doesn't really matter. So what we're doing now is we've got a team of dedicated curators who are looking at mutation actionability and looking across the literature for real detail on which mutations are actionable and what is their actionability, which mutation, which disease, which drug, with survival statistics coming out of that in detail, to make sure that we can feed clinicians high quality, useful, easily interpretable data to navigate patient treatment, because it's a complex world out there, and if we can make it much more simple and up to date, then hopefully we're doing a really useful job.
Yeah, that's sorry, I totally second that. One of the things that, as you mentioned, you have you run a large panel. There are variants that come in. I guess the example you used was a case where the scientist was spending 6 hours on trying to interpret that variant, and then you throw on top of that one of the key sources for doing that variant interpretation and making the recommendations of the treatment options would be keeping up with the clinical practice guidelines. And it's in some areas, those clinical practice guidelines, they change quite dramatically a few times a year. So in addition to that 6 hours to interpret that variant, they need to go read the non-small cell lung practice guideline, which is 200 pages, and digest it. That's one of the areas I think where these software tools are going to be really I think they are really valuable, and I think that's going to continue to be the challenge, which is to have the scientists on the curation teams and the knowledge modelers, et cetera, get that information, curate it accurately, put it in an application where the people can have access to it, and then finally make use of that. Make sure that the content and the decisions that are being suggested there, the clinical trial, recommendations and things like that are up to date and accurate, and so they can be reported out with a degree of confidence. And also making the subsequent information, easily accessible, because as detailed as our finding models are and the findings in HGMD and Cosmic are, they came from an article with many, many pages, and we can distill it down very well and use it effectively, but getting them to the references quickly so that they can, verify for themselves and report it confidently is, and that's, I think, why we do most of this, right? To do that Google Scholar search or whatever search they want to do, and make it. I guess we're down to 6 hours. It would have been considerably more than that before, but the complexity seems to be growing at about the same rate as our efficiencies do. So maybe we're just breaking even.
Yeah. So interestingly, by using a more modern approach with centralized database and depth, breadth, quality of information, I mean that lab did actually get down to an hour and a half.
Yeah.
With that gene panel. But Lisa, did you want to comment?
Yeah, definitely. I just think again, just having this experience, the scientists with more than 5 years, of experience doing the curation is just priceless, because they're able to really get to the key information very readily, and then they can, structure that content and then present it in a way that's just easy to assimilate, right? So when you look at HDMD and many of these other databases as well, it's not only that you have that human curation being done, that's saving a tremendous amount of time, but also that it integrates additional content sources. So in HDMD, there are several different databases that are integrated there, such as prediction modelers and dbSNP and others, that allows for you to get that context of the variant in HDMD directly without having to waste time going separately to all of these additional content sources. So I can really relate to the pain of, taking 6 hours or 7 hours to try to work up a case, and then just the tremendous time saver that you have, having all of that content just clearly presented to you, and content that you could trust in a database.
So, I mean, just to summarize here, what I'm hearing from all of you is really, where the knowledge base is going to really help is, first of all, centralizing, but very importantly, it's have the depth, breadth, and quality that you can trust, and it's up to date. I mean, those are the key elements of what's needed for a knowledge base that's going to deal with some of these challenges of volume and complexity of the information. I want to move down to helping the audience here as they're looking to acquire or adopt, embrace different tools, different software to support their interpretation. All three of you alluded to early on some of the way we curate information into those knowledge bases. Would you like to just describe what are the different approaches to curation? There's more automated, there's more sort of manual curation. And then, very important, I'd like you to sort of say what do you think are the positives and the negatives around this. One may be more machine-based, one may be more human-based, maybe a hybrid of both. Do you want to just comment?
Yeah, so I can start off just essentially, for HDMD, and I think I won't speak for the other databases here, but you do have this manual curation that is being done. And we're doing the releases quarterly, which again keeps the content up to date. It is really great to be able to, have scientists look directly at that literature, find that information. I think a strength of the manual curation is that, publications aren't perfect. So there are sometimes errors in the publication, either based on the genomic mapping that was done or perhaps a phenotype that was not reported accurately. And because you have a human doing that read-in and interpretation from the primary literature, they can actually reach out directly to the authors of the publication to clarify anything within that publication. And certainly our scientists at HDMD have at Cardiff University, they have done that on several occasions, clarifying information, and then authors would then update their publications. So again, before we are bringing it into our database, you're having that additional QC check so that you really are not then perpetuating that error down the line, right? So I think that's really a strength. I've heard of, several other vendors there, and I've used a couple before in my other roles before I joined QIAGEN. And I did find that you get a ton of data, but often it was the case that I was still trying to look for the needle in the haystack. So where it might seem really great to have more quantity, more of something typically sounds good, it can also still end up, draining some time because it's not necessarily the relevant or pertinent information that you need to interpret that case.
Yeah, definitely. I mean, I think, obviously there's completeness of, or, a large amount of data where there's a lot of false positives, a lot of false negatives that require validation, one route. The other one is just, more quality, trusted information. Simon, Shawn, do you want to comment?
Yeah, I think the emergence of text mining type technologies is very exciting. That can help curation quite a lot, although it's difficult to see it replacing it anytime soon. There are obviously people making text mining databases out of the literature, and we compare ourselves to them every now and again. And while there's a lot of information there, they're so full of noise, it's incredible. It's difficult to find the signal, and as Lisa said, the haystack is gigantic. And I think that technology will come to assist manual curation, but it's difficult to see, at least in the short and medium term, that it will replace it. But there's an additional thing that I see as a real value of manual curation is not just being able to clearly identify the valuable information and tease it out of this really complex language, is also what you might consider more unexpected points of information. So while we expect a curator to identify mutation and a disease to link together, there's a lot of information on a patient's gender, age, ethnicity, exposure, all of that information that lends itself very well to interpreting each of these mutations and beginning to put together more useful, more accurate patterns of the genetics behind disease. And I think our curators across the 20 years we've been doing this have been teasing out those data points, and now we have a database not just of millions of mutations against thousands of diseases, but also several million of these data points that say this mutation was observed in that disease, but also the patient presented in a particular way, or as a result of therapy, the patient reacted in a certain way. And you can look at sort of drug response, drug resistance, patient journey annotations that really give you much greater context around this information than simple linear patterns of mutations.
Sure.
Yeah, I agree entirely. I mean, I think any type of automation, we have a lot of our content acquisition workflow is automated. We don't start by querying PubMed and these sources, and we have code to do these kinds of things. And like any tool, I think that comes out, oftentimes there's where you think it may be someday, but then and it's could it replace curation of very simple concepts that are in articles? Probably. I don't know that we're there just yet. We'll have to learn it and test it. But as we talked about in the first part of this discussion, is as more and more patients are getting tested, and there are more and more therapies, and there's more and more finding the details that are in the in the various observations and getting them accurate and correct. You kind of get the sense that maybe ML and AI or NLP processes would be useful for the early case when there was only gefitinib as an EGFR inhibitor. But now that there are a multitude, it may get a little bit more difficult. So I think like any tool, being aware of how to use the tool properly, and then applying it appropriately so that you don't overstep that is always it's a balance point with any tool, and I think this one is the same way. The libraries that are out there and the accessibility to using these has gone down, and the bar is much lower now. But one thing also to remember is that a lot of these things rely upon training sets, and so that the content that we have, that high level content that's predominantly manually curated with all that detail and so forth, is going to serve to train the machine learning algorithms to try to extract it. So coming up with ways of integrating that into a manual workflow, I think is it's going to be similar to the more rule-based approaches, actually. But I think everyone who's worked on this type of thing for a while is aware of those challenges, and probably sees the same steps happening, only with different tools.
Yeah. I think, going back to some of the previous challenges, the complexity of all the interrelated information, the fact you're dealing with unstructured journals, publications. I fully agree that, AI and natural language processing is going to play more and more an important role in this, but I certainly struggle for, the immediate foreseeable future, how it could replace it. It's simply just because in science, we're so reliant on the context of a result. it can mean two different things. I remember years back, just looking at a term percent inhibition with a large pharmaceutical company, we came up with 23 definitions that we distinctly unique. And that's the size of the problem. And we use acronyms left, right, and center, which is another sort of challenge that we have here. So I think that human element of peer review, curating and putting that manual sort of review on top is so important to the quality. But ultimately, what's the impact of this? What does it mean, of having high quality data? I mean, do you guys want to, comment in terms of the impact to the actual person doing the interpretation, what you feel the results are?
Yeah, I think it can make a tremendous impact in terms of patient care, right? Because several of our labs, they're doing some complex testing, but also looking at rare disorders. Many times are impacting pediatric patients. So at the end of the day, we're all here working towards one end, I believe, which is to really impact patient care. So I believe, again, it's very important to have a database that is frequently updated with the content that you can trust, just making sure that you have the quality information. But there are several customers that have used HDMD to help them with prioritizing variants as well and determining what are likely the causal variants. And that can be really imperative when you're trying to, again, have a shorter turnaround time to be able to give an answer to a patient that's perhaps on a diagnostic odyssey. And I also want to add that, we've talked a bit about QCI Interpret and the QIAGEN knowledge base, and Interpret has a way of being able to incorporate both HDMD and COSMIC or some elements of it as well. So there are ways that, we can really match what is the best solution that is needed based on the customer needs.
All right, thanks, Lisa.
I think quality is a word I'm going to pick up on there and agree with Lisa that if you don't have a quality database behind the starting point of your work, it's very difficult to trust the consequence of that work. So quality is very much what all our processes work towards, and that's why, similar to Lisa, we have long-term curators with substantial QC processes and pipelines and everything to make sure that the data is correct and is well annotated and is easily accessible. And that forms the basis of really accurate reporting pipelines, target characterizations, and as Shawn said, you've got to make sure that your AI is trained in a really good fashion with trustworthy data. Otherwise, your AI is going to go all over the place. So it's about making sure that the quality is the best you can make it. It's absolutely top priority is the quality of this information.
Trust the information, absolutely.
Yeah, I everything the other two panelists said, I absolutely agree with, especially around quality, and and especially because we are in a clinical space, so quality is always important. Customers expect it. I think collectively the expectations of customers has gone up. Sometimes it seems like at a very sharp slope over short durations, but having the customers expect quality, and I think everyone here has mentioned that. So having that content and being able to use it to test and then do other things and open other doors is very valuable. But yeah.
And I'll go back to the slide that sort of provoked this conversation here, which is 45% of molecular pathologists, when questioned, said there was 7 out of 10 or less confident. And I think your guys' comments resonate. It's really about quality, being able to trust the data. It's taking that complex information and being able to use that to support those precisions, diagnosis there and interpretations. So I want to ask one final question here as we wrap up. My apologies. So, looking at our audience here who's come to learn about the knowledge bases and the different types and how they're curated, what's your advice to them in terms of what to look for when choosing a database to support their interpretation needs?
Yeah, absolutely. So, in terms of the interpretation, I would say you want to use a database that you are pretty certain that it has that reputation that the content has been vetted, right? So there might be databases that pop up, ever so often, but sometimes they don't have the longevity, they don't have that reputation. And I've unfortunately, encountered customers who their pipelines totally were outdated because they chose a database that was very new and it was not kept up to speed, essentially. So just ensuring that you have the quality, but I think also the reputation that goes with it, ensuring that the database is going to be updated frequently, because we do see, for hereditary cases that, every 6 months is a good rule of thumb to take a look at the variant classification to ensure that nothing has changed. And whether you're doing clinical testing or you're producing a publication, you of course want to make sure that you have the accurate information whenever you're putting those findings classifications on your report or your paper. So I think it is important to just make sure you have the quality there all around and really going with a database that you can trust, that has the reputation to back it up.
Sure.
Yeah, definitely, and especially in some of these spaces around the timeliness of the updates. So if you're reliant upon a workflow, if your workflow is integrated with a database and you're not able to get the information as quickly as is needed to do your reports, then you're going to have to come up with workarounds and things like that, and it's going to probably quickly eat away at any efficiencies that you have. So with the QCI interpret, the QIAGEN knowledge base, we're updating constantly. We roll out content updates to our applications roughly weekly. So when that new drug approval or a guideline update happens, you have access to that information from that reputable source rapidly, so that you're able to to pivot quickly on any new development or a drug approval that has occurred. Once the NTRK inhibitors, when the first NTRK inhibitor was approved, that obviously was something that for patients that had cancers where that would have been applicable, you want to be able to report that out quickly and use it in your standard workflow. So.
All right, Simon.
Yeah, and I'd agree with all of that. Quality, obviously, is something we've all agreed on en masse across this discussion. I think, in addition, does the information that you're looking at, does the database provide that data at high enough resolution? I think there are a lot of resources trying to describe this kind of information across high-level annotations. It's easy to see a range of mutations in lung cancer or blood cancer. But you only get meaningful interpretations when you're looking at very specific types of blood cancer, for instance. And those very specific types need to be almost the kind of thing a hematologist might see once or twice in a career. But by putting together large numbers of case studies across those instances, you get really clear pictures and very high resolution pictures of what matters genetically. And then, of course, standard FAIR principles apply. Is this data provided in good quality standardized annotations? Are you using standard genotypic genomic annotations? Are you using good vocabularies? And of course, is it easy to take that data and plug it into your workflow? As Shawn says, no one looks at millions of mutations in isolation. It's plugged into something else. Therefore, it's got to be annotated in a useful way to plug into something else. And the quality of the annotation and the interoperability of the data are vital when you're looking at databases of the scale we're discussing today.
All right, fantastic. So thank you to the panelists here. I want to sort of pull this to a conclusion here, but we set out with the topic powering the precision in precision medicine. I think the key that you've heard from the panelists here is the size of the panels, the whole genome, the complexity of the information here. Knowledge bases are going to become more and more important, and centralized access to high quality, trusted information is going to become more and more important as we start to scale in this world of precision medicine. If we're to get that 7 out of 10, 45% of people up into that high level of confidence, to deliver that precision medicine, there's got to be more adoption and use of these, high quality knowledge bases going forward. So big thank you for attending this webinar today. Thank you to the panelists. This concludes the session for today. Thank you.
Thank you.
Thank you. Thanks.